Welcome back to the science behind. Neocaridina shrimp are generally considered to be among the easiest freshwater invertebrates to keep and to breed. Give them a stable environment and a little bit of time, and it's not uncommon for a small company to grow into hundreds of shrimp. But despite that reputation, one of the most common questions in the hobby is still, why are my shrimp dying? And usually the answer you're given is that something is wrong with your water parameters, and while that might not be incorrect, it's rarely all that helpful because it's so incredibly vague. What parameter? Why does it matter? And more importantly, what is actually happening inside the shrimp that causes it to die when those conditions change? Because whether it's ammonia, copper, a failed molt, or something else entirely, shrimp don't just die because a number on a test kit is too high or too low. There's always an underlying biological reason. So in this episode, we'll take a closer look at some of the most common causes of shrimp deaths and the biological processes behind them. When we talk about shrimp reacting badly to water changes, it usually gets explained in very broad terms. Don't change too much at once, match your parameters, and acclimate slowly. But underneath all of that is one very specific biological system, which is called osmoregulation. Shrimp keep a fixed internal balance of ions, sodium, potassium, calcium, magnesium, chloride, and bicarbonate. Those levels don't really drift because they control nerve signals, muscle movement, enzyme activity, and the stability of the hemolymph. Everything that the shrimp does depends on that balance staying tight. The water around them has its own ion concentration. That is what we measure as TDS, GH, and KH. And because shrimp exchange ions through their gills and through the thin membranes between the plates of exoskeleton, the outside concentration directly affects the inside. There's no way for the shrimp to slow that exchange down, and it's continuous as well. So when the mineral content of the water shifts quickly, the gradient across those membranes also shifts immediately. The shrimp therefore has to adjust by pumping ions in or out through specialized transport cells in the gills. And those pumps have a maximum rate. They can only move ions so fast. If the change in TDS, GH, or KH requires a faster adjustment than the pumps can handle, the internal concentrations will start to drift. And once that happens, several systems in the shrimp will actually begin to break down. First, the sodium-potassium gradients weaken. Those gradients are what allow nerves to fire and muscles to contract. As they flatten, the shrimp loses coordination and becomes less responsive. Calcium regulation also becomes unstable, because calcium is tightly controlled and it affects muscle function and the molting cycle. Even a small shift can interfere with normal movement or the ability to complete a molt. Water movement across cell membranes will increase, and it's not dramatic, but it's enough to disrupt enzyme activity and raise the shrimp's energy demand. The shrimp will then start burning through its reserves, just trying to restore that balance. And the gills, which are the ones that handles most of the ion exchange, they become overloaded. When the gradient is too steep, they can't keep up, and the internal environment becomes progressively less compatible with normal function. And none of this will actually produce any visible symptoms. There'll be no discoloration and there will be no sign of external damage. The shrimp simply reaches a point where it cannot maintain stable ion levels anymore. Acclimation helps because it slows the rate of exchange down. It gives the ion pumps time to adjust their output as the external concentration will shift. Now, rapid changes in mineral content are one of the most common triggers for acute shrimp losses. Not because the final parameters are wrong, but because the transition happened faster than the shrimp could regulate. Toxicity is perhaps the straightforward category of shrimp deaths. These are substances that interfere with basic biological processes directly without needing a parameter swing or a secondary effect. When they are present, the shrimp system will fail because the molecules themselves disrupt essential functions. Let's start with ammonia. Shrimp rely on their gills for gas exchange and ion regulation. Ammonia crosses those same membranes easily. Once inside, it shifts the internal pH and interferes with the gradients that nerve and muscle cells depend on. Even low levels of ammonia can create stress because the gills have to work harder to excrete it, and that extra workload competes with the ion pumps that maintain internal balance. At higher levels, the pH shift becomes incompatible with normal enzyme function, and the shrimp loses the ability to regulate anything at all. Nitrite works a bit differently. It binds to hemocyanin, the oxygen-carrying molecule inside the shrimp, and reduces its ability to transport oxygen. The shrimp can be in well-oxygenated water and still experience internal oxygen shortages because the transport system is blocked. The gills respond by increasing ventilations, which increases ion loss, which adds another layer of stress on top of the oxygen problem. Now, copper is a direct cellular toxin. Shrimp use copper in their hemocyanin, but only in a very controlled form. Free copper ions interfere with proteins, they damage gill tissue, and they disrupt mitochondrial function. The gills are the first point of contact, so they take the hit immediately. Reduced surface area, impaired ion transport, and increased permeability. Once the gills are compromised, everything else will follow quickly because the shrimp can't maintain internal stability. Chlorine and chloramine damage tissues through oxidization. The gills are, again, the primary target. Oxidative damage reduces their ability to exchange gases and ions, and the shrimp loses regulatory control long before the exposure will then become lethal. Chloramine is especially persistent, so even in small residual amounts can cause cumulative stress over time. Across all of these toxins, the pattern is the same. They interfere with processes that the shrimp cannot compensate for. Fish have more internal buffering and also thicker protective layers. Shrimp don't. Their gills and membranes are thin, exposed, and they're constantly active, which makes them more sensitive to anything that interacts directly with cells or proteins. So the toxicity isn't very subtle, and it doesn't require a parameter swing or a slow buildup of stress, but instead it's a direct interaction between a molecule and a biological system, and once that interaction starts, the shrimp will then have very few defenses. Molting is the single most demanding process a shrimp has to go through. Every growth cycle will depend on it, and every molt carries a real risk because the shrimp has to rebuild critical structures while its defenses are at their weakest. The process starts long before the actual shell will crack. Shrimp store calcium and other minerals in small internal reserves called gastroliths. These reserves are dissolved and reused to harden their new exoskeleton after the old one is shed. If the mineral balance in the water is off, too low, too high, or maybe inconsistent, the shrimp can't build those reserves properly and the new shell won't harden fast enough. The actual trigger for molting is hormonal. A drop in molt inhibiting hormone allows the next stage to begin, and once that signal is released, the shrimp is committed. There's no way to pause or reverse it. The old exoskeleton will separate from the body, the new one will form underneath, and pressure will build until the shrimp forces its way out. During the molt, the gills, joints, and membranes are exposed. The shrimp can't regulate ions as effectively, and it can't defend itself from rapid changes in TDS, GH, or KH. Any instability in the water at this moment has a much stronger impact than it would at any other time. One of the most well-known failure points is the white ring of death. It appears when the old and new exoskeletons separate incorrectly, leaving a visible white band around the shrimp's midsection. The band marks a structural break where the shrimp can't generate enough pressure to complete the molt. Once it reaches that stage, the shrimp is usually unable to free itself. Failed molts can also happen without visible signs. If the new exoskeleton doesn't harden quickly enough, the shrimp remains soft for too long. Muscles will then not anchor properly, movement will become difficult, and the shrimp cannot maintain normal function. Calcium imbalance, low GH, or disrupted osmoregulation all contribute to this. The period immediately after molting is just as risky. The shrimp is soft, its gills are still adjusting, and its internal ion balance is more sensitive than usual. Even small fluctuations in mineral content or pH can push it past what it can compensate for. But molting is essential. It's how shrimp grow, repair damage, and maintain their exoskeleton. But the combination of hormonal commitment, mineral demands, and temporary loss of structural protection makes it one of the most vulnerable points in their entire life cycle. Not every shrimp death can be traced to a specific parameter or a single toxic event. A large portion of losses come from slow, compounding stress, the kind that will weaken the shrimp over time until something relatively minor becomes fatal. Shrimp have a simple but still effective immune system. It relies on hemocytes, antimicrobial peptides, and a set of enzymes that break down pathogens. These defenses work well when the shrimp has enough energy and stable internal conditions. but they don't have much reserve capacity. Anything that increases metabolic demand or disrupts ion balance reduces the resources available for immune function. Bacterial infections will then take advantage of that. Most of the bacteria involved are already present inside the system. They become a problem when the shrimp's defenses fall behind. The gills, gut lining, and the soft tissue around the joints are usually the entry points for bacteria, and once that bacteria gets past those barriers, the immune system has to contain them quickly. If it can't, the infection will spread throughout the hemolymph and it will overwhelm the shrimp. Parasites, they follow a similar pattern. They rarely cause immediate death on their own, but they drain energy, damage tissue, or interfere with feeding, and the shrimp will then slowly lose the ability to keep up with normal demands. By the time symptoms become visible, the decline has usually been happening for a while. Chronic stress is often the underlying factor that makes these problems possible. Stress in shrimp isn't psychological, it's biochemical in this sense. It shows up as increased iron loss, elevated metabolic rate, and reduced efficiency in the gills. Anything that forces the shrimp to spend more energy on basic regulations leaves less available for growth, repair, and immune function. Low-grade instability in TDS, GH, or KH, minor temperature swings, overcrowding, or constant disturbance all contribute to this. None of them are lethal on their own, but together they shift the shrimp's energy budget away from long-term maintenance. Over time, the immune system becomes less responsive and infections that would normally be contained will then start to spread. Symptoms often appear late because shrimp compensate quietly and they don't show obvious signs until the internal systems are already failing. By the time you see lethargy, discoloration, or unusual behavior, the underlying decline has usually been progressing for days or even weeks. And shrimp rarely die from a single cause. It's usually a combination of a small parameter fluctuation, a minor injury, and perhaps a bacterial load that was already present. And the shrimp whose energy reserves were already stretched thin. The final trigger will obviously look sudden, but the decline leading up to it is almost always cumulative. Some shrimp losses come from internal factors that aren't visible from the outside. Neocaridina or caridina shrimp don't live that long, and as they age, their ability to regulate ions, fight infections, and complete molts gradually declines. The system that keep them stable lose efficiency, and small stresses have larger impact. Genetics will also play a role. Selective breeding, as it has been happened for many years, reduces genetic diversity, and with it, the resilience that wild populations rely on. Some individuals will carry weaknesses that only show up under mild stress. Irregular mold cycles, reduced immune responses, or poor mineral handling. And those issues aren't obvious until the shrimp is already struggling. There are also internal failures that don't leave external signs. A disrupted mold signal, a metabolic imbalance, or a minor infection that progresses faster than the immune system can control. By the time you will see behavior changes, the decline is, as mentioned, well underway. Losses from aging, genetics, and internal failures are also normal in closed populations, and they don't point to a major problem in the tank, but they're parts of the biology of the species. So when we ask why shrimp die, the answer isn't a single parameter or a single mistake. It's a combination of systems that all depend on stability, ion balance, gill function, molting, immune response, and the limits built into their genetics. Osmoregulation makes them sensitive to change, toxins interfere with basic cellular processes, and molting pushes their physiology to its limits. And long-term stress or internal weaknesses can turn small problems into larger ones. Understanding these mechanisms don't prevent every loss, but it makes the patterns clearer. It separates normal mortality from the issues that need attention, and it gives us a better sense of what's actually happening inside the tank. And it gives us a better sense of what's actually happening inside the shrimp when things go wrong. Thanks for watching this episode of The Science Behind. If you enjoyed the video, why not subscribe? If you have any questions or suggestions for the topics, leave them in the comments. I try my best to read every last one. Bye for now.